Let's move on now and talk about this process called translation. From the central dogma, remember, RNA is used to make protein, and it's ribosomes that are going to do this work in the cell. The mRNA, the messenger RNA molecule, is a string of nucleotides, and those nucleotides all have a base. So a messenger RNA molecule has a particular sequence of bases in it, A's and U's and G's and C's. The ribosome is going to attach to the messenger RNA, and it's going to read these bases. It's going to read them like a code, and it's going to build a string of amino acids from that code. That's what a polypeptide is. The polypeptide will then fold up and become a functional protein. Now, the ribosome doesn't read one base at a time. Instead, it reads groups of 3 bases. We call those groups of bases codons. Now, how does the ribosome know where to begin reading this protein code? The answer is it looks for and it binds to the mRNA just before the first group of 3 bases. The first group is called the start codon, and the start codon is the same in every piece of messenger RNA for every living cell. Doesn't matter if it's a microbial cell or another type of cell. It doesn't matter if it's a prokaryotic cell or a eukaryotic cell. For every protein that needs to be made, the ribosome is going to begin at the start codon. Just like we saw for transcription, there are going to be 3 phases to translation. And lucky for us, they have the same names. There's going to be an initiation, then an elongation, then a termination. Initiation is the phase where the ribosome first comes in and binds to the messenger RNA, just before that start codon, just what we call upstream of the start codon. It lands there, and then it begins this process of traveling down the messenger RNA and reading each group of 3 bases, each codon, and decoding that codon so that an amino acid can be chosen for that location according to the code. The ribosome has to physically move, just like the RNA polymerase enzyme had to physically move down the length of the DNA. The ribosome is going to move down the length of the messenger RNA. We call this moving translocation. The ribosome will travel downstream, and it will expose one codon at a time so that an amino acid can be matched with that codon according to the genetic code. Once that amino acid is in place, the ribosome will translocate to the next codon. It will expose the next codon. An amino acid will be placed in that location. The 2 amino acids will be bound together with a covalent bond. And remember, we call those peptide bonds in protein. Then the ribosome will translocate, move to the next codon, and the process will repeat. This process, where each codon is read, each codon is decoded, is called elongation. And this process brings in another player to this gene expression process. This other player is transfer RNA, or tRNA. transfer RNA does the work of bringing in the correct amino acid for each codon. The ribosome exposes the codon. The ribosome is going to do the work of attaching one amino acid to the next, but the transfer RNA, the tRNA, is literally carrying in the amino acid to be added into that chain of amino acids. Now, elongation will continue all the way down the messenger RNA until the ribosome encounters a stop codon. Remember, there are 3 codons in the genetic code. I have them written out here. You don't have to memorize the bases in the stop codon or anything. The genetic code table is available to you at the tip of your finger just by Googling genetic code table. So you can always find which combination of bases make up the stop codons. But when the ribosome encounters a stop codon, that's going to bring us into the 3rd phase of translation, which is termination. The reason that translation terminates when the ribosome hits a stop codon is because there is no amino acid that matches these stop codons. These stop codons don't code for any amino acids. So the process will end. Termination will occur. The protein has been made. It's been built. So this molecule called transfer RNA has a really important job. It binds to amino acids and it carries them in according to the ribosome, according to which codon is being exposed in the messenger RNA. The tRNA are multiple in number. In other words, there's a different tRNA for each of the amino acids. We haven't talked too much about amino acids, but there are 20 commonly occurring amino acids. So we need a different tRNA for each one of those. Remember, there's a code that matches each amino acid. There is a codon. There's a group of 3 bases that is the code for each amino acid. The tRNA molecule has to be able to read that code, so we know we're matching up the correct amino acid for the correct codon. And the way the tRNA interacts with the codon is by binding to it. And again, this process takes advantage of that complementary nature of nucleic acid. The group of 3 bases in the messenger RNA is called a codon. At one end of the tRNA molecule, there's going to be 3 bases. That's called an anticodon. There is an anticodon for every codon. So only the tRNA that has the correct anticodon can come in and bind to each of the codons in the messenger RNA. There's only one tRNA that will match each codon and be able to bind to it. And attached to that tRNA, there's going to be an amino acid. Now remember, every living cell on planet Earth uses DNA as its genetic material. Every living cell on planet Earth uses ribosomes to do translation. Every living cell uses the same genetic code. And the genetic code is extensive. You've seen the table. It has 64 codons listed in it. 64 codons that will encode for 20 amino acids. That doesn't seem right at first, but remember, there's room for error in the genetic code. There's a little room for error. We give that a name. We say that the genetic code is degenerate. It sounds terrible, but what that means is there's more than one codon for each amino acid. Remember, we said there are sometimes 2 codons, there are sometimes 4 codons that all code for the same amino acid. And the reason that that's important is because it allows for some mutations to occur in nature without disrupting the protein building process. It allows for some mutations to occur without changing the protein that will be made from that gene. Remember too that it's the 3rd position in the codon that is very wobbly. It's the 3rd position that can change quite a bit, and we'll still see the same amino acid placed in that position without changing the protein. On this slide, we can see the genetic code table. You've seen this before. This table shows us every possible combination of 3 bases that can be found in messenger RNA. Remember, there are 4 bases, A, U, G, and C, and the ribosome is going to read them as groups of 3. So we're looking at every possible combination of 3. And you can see in the table that next to groups of codons, there's an abbreviation, a 3-letter abbreviation, that stands for a particular amino acid. So for example, the codons AAU and AAC both code for an amino acid called asparagine. Over here, we can see a group of 4 codons, CGU, CGC, CGA, and CGG. All 4 of those codons code for the amino acid called arginine. This is an example right here of this wobbliness of the 3rd position in a codon. Remember, there are only 4 bases, and basically what you're looking at here is that the 3rd position can be any of those 4 bases and still code for the amino acid arginine. So a mutation that changes this 3rd position is not really going to matter, because we're still going to get arginine placed at that position. We're going to get the same protein made. Remember, there are different kinds of mutations, and we talked specifically about point mutations. We said point mutations involve a single base being changed in the DNA. Sometimes that change, that swap of one base for another, will change the codon enough that it will result in a different amino acid being placed there during translation. If even one amino acid is out of order in a protein, even one, that protein will fold differently. And if it folds differently, it won't function the same. The disease called cystic fibrosis is an example of a substitution mutation. There's one base that's not in the right place, just one. And that change in that one base changes a codon in the messenger RNA in that one position that leads to a change in which amino acid gets placed there. And the protein that gets made doesn't function correctly. And the person ends up with a whole list of diseases that comprise this syndrome that we call cystic fibrosis. Sometimes when a base is substituted for another, you wouldn't even know it, because the change in the base, the substitution in the base, doesn't change the codon enough. And you end up with the same amino acid placed in that location, so the protein is the same. Those are the silent kind of substitution mutations. Sometimes the change in a single base creates a stop codon where there didn't used to be one. That's a nonsense mutation. Remember, if the ribosome encounters a stop codon, it's going to stop. It's going to stop building the protein right at that location. Maybe the protein should have had 1,000 amino acids in it. Maybe the stop codon should have been at position 1001. But a mutation occurs in the DNA, and it results in the production of a stop codon in the messenger RNA at location 30. When the ribosome hits that stop codon, it's going to stop. And you're going to get a protein that has 29 amino acids in it instead of 1,000. That protein is going to be terribly affected by that. We call those nonsense mutations because the protein is often just a nonsensical form of itself. Remember, frame shift mutations are also pretty massive in their effect. Frame shift mutations are different from substitution mutations. In a frame shift mutation, an extra base is added, or a base is completely deleted from the DNA. What's going to happen is the groups of 3 bases are all going to shift by 1. They're either going to suddenly shift because there's been an extra one added in, or they're going to shift because one has been taken out. In other words, every codon past the point of that frame shift mutation will be affected. Every amino acid past that point, at least potentially, could be affected. And you often end up with a very changed protein.